Hello everyone, I'm Fiona Murdoch. Thanks for coming to this research update on the Mallee Looper caterpillar, hosted by Friends of Mallee Conservation in partnership with Wildlife Unlimited. This Victorian research has been funded by the Future Drought Fund and Natural Resources Conservation Trust, but I know there's many South Australian people in this webinar, so I'll also be providing an update on the South Australian research that's underway. Much of our research took place on the lands of the first people of the Millewa Mallee, being Latchi Latchi and Ninte traditional owners. So I pay my respects to elders past, present, and Aboriginal elders who may be with us today, and acknowledge traditional owners of the various lands where we all live. So we'll start today with a background into the Mallee Looper caterpillar. What is it and why do we care? We'll discuss the current outbreak and outline our progress in filling some key knowledge gaps. Following the presentation, which will run for about 30 minutes, there'll be an opportunity to share your observations and ask any questions. So as you think of things, just pop them in the chat. We'll have a discussion at the end. So this is the Mallee Looper. It's a caterpillar of a native species of moth that's usually pretty common across southeastern Australia. The moths in the genus Arhodia, but the taxonomists are still working on describing all the species in that complex, so we don't actually have a species name yet. And it's called a looper because of the way it moves. It's got legs at the front and legs at the back of its body, so it loops along rather than crawling. Now, over the past few years, we've built up a bit of an understanding of the life cycle of the Mallee Looper. And when I say we, I mean an interstate collaborative network called the Looper Groupies. Now, this is a loose collection of ecologists and rangers, volunteers, entomologists, and academics who've been quietly working away to unravel the Mallee Looper mystery. And nearly all of what we know about the Mallee Looper has been contributed by this group. In addition to the Looper Groupies, there's so many citizen scientists who've been engaged through this project, and particularly through iNaturalist, and these eyes on the ground have been really critical to get us where we are today. So this is what we know about the life cycle of the Mallee Looper caterpillar. The moths typically emerge in spring and they mate and lay eggs, which hatch into tiny caterpillars about 10 days later. The caterpillars grow from about 4 millimeters long to about 5 to 6 centimeters long, and they're eating all the time. And it's the larger caterpillars that are the hungriest, and they're the ones that can cause the defoliation of mallee trees. After around a month of being a caterpillar, they climb down from the trees and they pupate underground over winter. Now, the main reason we're interested in the Mallee Looper is because the caterpillars eat the leaves of our Mallee Eucalypt trees. And over the past 5 years, they've been continuously superabundant in parts of the Victorian and South Australian Mallee. And when there's lots of Mallee Loopers, that can be thousands of caterpillars on a tree. They can completely defoliate areas and it looks like a fire has been through. So none of the trees in this photograph have any leaves left on them after a looper outbreak has been through. Now, this is the area that's most affected. It's the Mallee of Northwest Victoria and adjoining areas of South Australia. Our research is in Northwest Victoria and it's been around Murray Sunset and Hattah-Colquhoun National Parks. Most research in Australia has been, in South Australia, has been within the Living Landscape group of conservation properties. So in particular, the Australian Landscape Trust, Kelpram and Taylorville stations, but also the Glupot Reserve, you can see there, managed by BirdLife Australia. Now, Glupot's really important in this story because that's where severe looper impacts were first noticed about 5 years ago. And unfortunately, the trees at Glupot have been hit hard by loopers year after year. And we know that mallee trees are really tough and they can regrow after fire, and they can certainly regrow their leaves after defoliation by a mallee looper. But several years of repeated defoliation and some of the trees have actually started dying or they're in extremely poor health. It's unclear if it's the Mallee Looper alone or if it's the looper combined with drought that's causing tree deaths. And it's not widespread, but poor tree health is really evident across much of the Glupot Reserve. So tree death's obviously the most significant and long-lasting impact of defoliation by loopers, but in the shorter term, we're also seeing things like reduced flowering and seed production because the defoliated trees are allocating their resources to regrowing leaves and they don't have anything left for reproduction. And this obviously reduces the availability of nectar for insects and birds, and it also reduces seed production of the trees. No leaves means there's reduced food available for other foliivores. I've really noticed that it's hotter in looper areas because there's no shade, and there's also less leaf litter on the ground. And these sorts of effects could be really significant for species and communities like threatened mallee birds. On the flip side, the moths and caterpillars could be a bonanza for birds and other things that like eating them. And even potentially some understory plants might benefit from increased access to light. So we've always got to think about both sides of the coin. Now, the history of the current outbreak is that we had some observations of defoliation by looper caterpillars in 2006 and then also 2010-11. But at the time, we couldn't determine the species of caterpillar responsible. In January 2022, Mallee loopers became abundant in Victoria and South Australian Mallee, and by the following year, 150 to 250,000 hectares was believed impacted. So that's when we finally gave the culprit a name, and it was identified as a caterpillar of a native moth in the genus Arhodia. Now, since then, outbreaks have continued annually, and glue pot reserve in South Australia has been hit year after year, but there's been patchy outbreaks that have been spreading south and east from there. So that's the background. Obviously, one of the key questions when you have an outbreak like this is, has this happened before? Is it a natural thing or something new? Has something gone wrong and do we need to control it? So I'll touch on that, and I'll also provide an update on how we're going with some of our key knowledge gaps. So has it happened before? Can't say for sure, but there is some suggestion that an outbreak occurred in the early 1930s and also the early 1960s. We don't obviously know if it's the same Mallee Looper species, but there were several newspaper articles in January 1931 that discuss a plague of looper caterpillars in northwest Victoria, and that description sounds really familiar. The place mentioned, Benoona, is just down the road from me, and it's also been heavily impacted in the current outbreak. Then in the 1960s, there's a research paper that refers to a plague of leaf-eating caterpillars, and that's supported by the recollections of local naturalists who talk about large areas defoliation along the Honeymoon Hut Track. Honeymoon Hut Track is in Murray Sunset National Park, and again, this is an area that's been severely affected in the current outbreak. So we do have some anecdotal evidence that we might be looking at something like this, an outbreak that's crossed a threshold where we're seeing lots of damage, and hopefully the looper will naturally return to lower levels of abundance and damage as an endemic part of the system, and perhaps it will pop up again in another couple of decades. That's the fingers crossed approach, but really we don't know, and we do need to be prepared if we're wrong and this looper outbreak becomes really prolonged. Now, we don't have many examples of native outbreak caterpillars in Australia, but overseas there's several. They've done a lot of research into them. This is the native spruce budworm. It kills trees in spruce and fir forests. And interestingly, the severity and the frequency and the extent of outbreaks is believed to be increasing due to climate change and forest management practices. So that's something for us to think about with the Mallee Looper. Now, with the spruce budworm, the theory is that outbreaks start when external factors surpass the natural control agents. Hotspots erupt and then they spread to other areas. And this possibly applies to the Mallee Looper also. Because the Mallee Looper, most of the caterpillars would normally die of natural causes, predators, pathogens, parasites, bad weather or whatever. But you can see that if we dial back any of these, it increases the number of caterpillars surviving to become moths, and that's how the outbreak spreads. So our working hypothesis with the Mallee Looper is that an abundance of fresh growth on the mallee trees, followed by a mild summer, that allows the looper numbers to build up. So fresh growth on mallee trees, that occurs after fires. It also occurs after good spring rainfall following drought conditions. And it also appears after looper defoliation in previous years. So that can actually kick off a cycle of defoliation. But we have by no means really fully understand what's going on with predators and parasites and pathogens in this case. So do we need to control the Mallee Looper? So absolutely, in some areas, spruce budworm outbreaks are actively controlled by spraying with a targeted pesticide. They use what's called an early intervention strategy. They detect and treat emerging hotspots really as quickly as they can. But to get to this point, the spruce budworm people have a cost-benefit framework. They know when it's worth acting, and they understand the economics and environmental impacts. They understand the characteristics of susceptible areas where the hotspots are likely to occur. They've got robust monitoring techniques, and they know what the thresholds for damage are. And they've got targeted control techniques. They're targeted to be effective and to minimise non-target impacts. We don't actually have any of these things for the Mallee Looper, so we're literally years away from even thinking about control. But it does mean that we need to chip away at all of our knowledge gaps so we can get to the same level as understanding as research knows about the spruce budworm and other outbreak caterpillars. So where are our knowledge gaps? Obviously the key one is giving this guy a name. It's been worked on, but the issue is that to name the Mallee Looper, we need to review the entire genus. So we're pinning moths and sending samples of caterpillars to the entomologists involved in this and assisting with that work. One of the issues is that there's huge variation in the appearance and even the size of moths. So you can see the patterning is quite different. And in the lower right there, you can see two moths side by side that are completely different sizes, and they're all Mallee Looper moths. Another gap is understanding the impacts and focusing on the mallee trees. So Australian Landscape Trust Calparum are monitoring 250 trees every month and assessing the flowering and fruiting of those trees. The preliminary results there are suggesting that heavily defoliated trees, it really does reduce subsequent flowering and fruiting. Also at Glue Reserve, the rangers there have the longest data set for annual trapping of moths. They've been really incredible over the last several years, trapping moths at Glue. But Glue also hosts University of Adelaide interns, and they conduct annual surveys of permanently tagged trees, recording the levels of defoliation and regrowth. So the red bars in this graph are the percentage of trees that are assessed as having more than 75% of the canopy eaten by loopers. So that's a huge amount of defoliation for a tree to withstand. And you can see how common those impacts were in 2022-23. So what did we do in this study? We added some more information to the detail of the Mallee Looper life history. We monitored moths and caterpillar abundance in the bush, and we did some work trying to identify where susceptible habitats might be, so areas that might serve as early warning of outbreaks or hotspots. So we measured the size and counted the eggs laid by 50 moths. Not surprisingly, larger moths lay more eggs. Most moths laid between 400 and 700 eggs, but the record was nearly 1,000. We also observed these eggs to hatching. We found it takes 10 days for the eggs to hatch, and pretty much 100% of them hatched. So there's no signs of parasites or pathogens affecting the egg stage. It's pretty tedious doing this sort of counting, but it's important knowledge. In nature, these eggs are laid at exposed locations on dead twigs at the tops of trees, and there can be over 100 egg masses in a single tree. There's a looper groupie in South Australia named Mark Hura, who set up the scaffolding you can see in the top right. So it's in his property where it's been severely impacted by loopers over several years. And this year he figured out why the moths lay their eggs in such exposed locations. And it's to aid the dispersal of the tiny caterpillars. After they hatch, the caterpillars hang from silken threads and then they float away on the wind. And that's a way for them to disperse into new habitats where there might be some fresh leaves for them to eat. So next we reared the caterpillars, and we were really interested in the growth rate, how fast caterpillars grow, and the stages that a caterpillar passes through. So as a caterpillar grows, it goes through discrete growth stages that are called instars. The caterpillar skin can stretch a little to allow growth, but eventually it gets too big for that skin sausage, and it has to molt the entire outer cuticle so it can keep growing. And underneath is a slightly larger skin for it to grow into. But because the head capsule is more rigid than the skin, it doesn't stretch as the caterpillar grows. So in effect, there's one helmet size for each instar. So we measured the head capsule width of 240 caterpillars and related this to body length and caterpillar age. And we got these really neat relationships showing that there's 6 stages or instars. And now we can use this relationship to predict when the larger caterpillars will be present in the bush, about 35 days after we see peak emergence of moths. And we can also estimate the age of a field collected caterpillar from its head size. And that's much more accurate than just using length. So these will be pretty useful ready reckoner charts. Now, I just wanted to show you these photos because I think pupation is the most amazing transformation, going from a caterpillar to a moth via this alien stage. But the Mallee Looper caterpillars, after they've eaten their fill, they descend from the trees and they pupate about 10 centimetres below the soil. The pupal stage does seem to be a bit vulnerable to predation because they spend the entire winter underground as a pupa. So things like white-winged choughs dig them up and eat them. I've noticed in some areas there's extensive digging by echidnas and also pigs in loopered areas. So these are potential predators as well. Through this study, we've been able to add a bit more detail to this Mallee Looper life cycle. So that's been pretty helpful. Now, along with rearing caterpillars in the office, we also used a couple of techniques to monitor what was going on in the bush. This year, we set up moth funnel traps in and around Hattah National Park, and we wanted to do that to monitor the size and the date of peak moth emergence. So the moths are attracted to the UV light in the trap, and they fly into the transparent veins, and then they fall through a funnel and they're collected in the bucket. And we did this every night for 2 months. The moths are not killed in the bucket, and we also get lots of bycatch of other creatures. So it's important that we open the traps early in the morning. We put the mesh bags in the fridge for a little bit to slow them down and then count the male and female moths. My record was 370 moths in one trap. So yeah, it can get pretty tedious counting those. And what we found is that there's way more males, which is the blue line, than females, which is the orange line. And at some sites, this sex ratio is reversed, and we don't know why. That's another knowledge gap to look into. And across all of our sites, there were 2 common peaks of emergence around mid-November and mid-December. And amazingly, the size and timing of these synchronized peaks of emergence was virtually identical to the trap data in Gluepot, South Australia. In the past years from the Gluepot trapping data, we've also noticed that there seems to be a correlation between rainfall events and this emergence of moths about a fortnight later. This year it was less clear because the rainfall was really scant. We also designed these do-it-yourself moth traps, which are based on the fancy moth traps, but just a lot cheaper, about $100. We gave these away to citizen scientists and found from their trapping at an additional 11 sites that the Mallee Looper moths were found right across the Mallee in all sorts of habitats, urban backyards, riverine woodlands. On this site, we didn't trap moths was patchyollic, which is right down in the south where we haven't seen any damage by loopers. So after the moths came the caterpillars, and we monitored abundance of caterpillars with a beat sheet, basically hitting trees with a stick and counting how many caterpillars fall out. And this works when the caterpillars are young, so the smaller instars, but it doesn't work with the larger caterpillars because they hold on too tight. But it is a simple method that can be used to quantify how many caterpillars are at a location. What we found this year is that there were a few caterpillars early on, but these quickly petered out, and by mid-January, we couldn't find any caterpillars in the bush. And the glue pot ranges in South Australia reported similar things. This result was also reflected in our damage assessments. We did baseline damage assessments prior to the loopers emerging, and then repeated them in February when we would have expected to see damage caused by the caterpillars. Only this year there was no damage, and in fact sites that had been previously defoliated were pretty much recovered. So we've surmised from this that the caterpillar stage, particularly the early instars, might be vulnerable to really hot weather because we did have some pretty serious summer heat waves this year. One thing we did note with the damage assessments is that different trees are affected differently. In areas like Blue Pot, where the outbreak's been going for years, all habitats and all trees are affected. But in Hattah National Park, where the outbreak's just starting, as much more recent, we thought we might be able to identify susceptible habitats where outbreaks start. So we looked at 3 types of mallee, Eucalyptus gracilis and Eucalyptus oleosa. They're both narrow-leaf species, but gracilis is often totally defoliated out in the bush. And then we compared those with Eucalyptus jumosa, which has really thick, coarse leaves. And that appears to be less preferred by the caterpillars. So we fed these different species to native caterpillars and that were in captivity, and we found something a little bit different to what we expected. The newly hatched caterpillars must have access to fresh growing tips of leaves. So if you feed them mature leaves, they die. Well, they just can't feed, so they starve to death. But it doesn't matter what species of eucalypt you feed them. They're polyphagous, so they can eat a variety of Mallee Eucalypts as long as they've got access to those fresh growing tips. When the caterpillars grow up a bit, you can feed them mature leaves. And we did have some preliminary observations that when given choice, the caterpillars would eat more of the Eucalyptus gracilis than some of the other species. The sample size was a bit small to be definitive by that point, though. We did try to have food preference trials out in the bush as well. We contained caterpillars in these organza party favour bags on different tree species, but we had 0 survival. And again, that was possibly due to those heat waves we experienced this summer. We've got some work underway at the moment using remote sensing to detect looper outbreaks. What we're looking for is a decline in vegetation greenness using an NDVI index. And we're looking at that, comparing between early spring and late summer. And we have been able to use this to detect affected locations. These are often heavy red-brown loamy swales, and they're often dominated by Eucalyptus gracilis. So we'll be expanding this remote sensing work over the next few months, because one of the things that raises the question is it the location in the landscape? So the swales obviously have a bit more moisture in them, so perhaps more fresh growth, or is it the species that grow in those swales like Eucalyptus gracilis that makes that habitat susceptible? So by February, we were feeling pretty happy that most areas appeared to have escaped looper defoliation this year. But then we had the big rainfall event at the start of March. In Victoria, Mallee, we had about half of our annual rainfall in a couple of days. And about a fortnight later, the Glue Pot Rangers reported that they'd recorded over 1,300 moths in a single trap in one night. Here in the Victorian Mallee, we also noticed high numbers of new egg masses appeared in the bush. So this was totally unexpected to have an emergence of moths that late. And it really does strengthen our theory that the moth emergence from the pupa is linked to rainfall. But yeah, as I said, we didn't expect moths to be emerging in autumn. What it does tell us is that maybe not all the pupa hatch at once or the moths emerge from the pupa at once in spring, and some of the pupa are left in reserve and can appear following later rainfall events. By mid-May, the loopers from the March emergence the looper caterpillars had reached full size and they've just started to pupate. Defoliation's been patchy, but it is certainly much more evident than it was in February this year. It's mostly on new growth that appeared after the rainfall event. And some areas have been hit hard with entire trees defoliated, but it's not the extensive areas that we saw in 2023. We have noticed that the cooler weather appears to have slowed the caterpillars down, so their growth rate is slower. They're taking about 2 weeks longer to reach pupation than caterpillars from the December emergence. And this, combined with cooler and wetter weather, appears to have allowed some natural biocontrol agents to appear, which is pretty exciting. Some of the caterpillars appear to have succumbed to pathogens. These look like just limp caterpillars hanging by their anal claspers. This entomophora has probably caused the cauliflower-like growths in the centre caterpillar, but there's also bacteria and viruses that could be responsible for the deaths. We've observed lots of cocoons from this tiny parasitoid wasp. Now, it lays its eggs into the second instar caterpillars, and then the larvae emerge from the bodies of the fifth instar caterpillars, which are alive at the time, so that's just totally gross. The larvae from the wasp larvae form these cocoons, and then the caterpillars are tricked by whatever means into becoming the bodyguard. So they actually look after the wasp cocoons until the wasps hatch. It's such an interesting life history. There's also a predator poking about, Schellenberg's soldier bug. It's one of the most common predatory insects in Australia. Both the nymphs and the adults feed on caterpillars. They stab them and they insert 2 tubes. One injects saliva into the caterpillar and one sucks up the partly digested caterpillar juice. And you can see these soldier bugs still with their mouth parts injected into a caterpillar that's looking quite deflated. Now, none of these individually at the moment are enough to cause the Mallee Looper population to crash, but it's really good to see these biocontrol agents building up. So, a summary. There are lots of mites on this slide, but the one thing that's absolutely for sure is having an informal collaborative network like the Looper groupies and also citizen scientists has been the only way that even this small amount of knowledge has progressed. There are lots of areas for future research. We have some answers, but there are more questions. And these information gaps can be filled by anyone in this meeting. And the only way they will be filled is by interested individuals, by you, so like you. So if you think you might like to contribute to understanding the Mallee Looper, please get in touch and we can share some more detailed information and you can join the Looper groupies too. So yes, thanks to the Looper groupies who have shared their knowledge with me so freely, and that's facilitated this research. And also thanks to our funders, the Future Drought Fund and Natural Resources Conservation Trust. Thank you very much.